Well, so good morning, good afternoon, and whatever you are. And my name is Lucia Lohman, and I currently serve as president of the International Association for Plant Taxonomists. And it's really a great pleasure to see all of you here. Today is our third webinar of the series, and it's kind of exciting to see people from all over the world, actually. Several familiar names, and very, very happy to see you all here. So I think the IAPT, you all know that it's really a global hub that facilitates taxonomic, systematic, and nomenclature research on algae, fungi, and plants around the globe. We were established in 1950, which makes this year a super exciting year as we are celebrating our 75th anniversary. So please be sure to stay a little bit to the end because we are going to have a special toast and to celebrate this special occasion. This is also important time to renew membership, so stay tuned for our newsletters and emails and make sure to continue to be part of this hub. The IAPT is really your association, so please also reach out if you'd like to be more engaged and to participate. We are really working on a series of training workshops and webinar series, in addition, of course, to taxon that we publish and the photo context. And we really would like to have a series of new initiatives next year. So please reach out and let us know how you would like to participate. Today is a very special webinar, wonderful speakers, and I will let Dr. Dirk Albach and Gulzar Khan introduce the speakers. But I just would like to welcome the two organizers of this webinar. So Dr. Dirk Albach, he is the managing editor of Taxon, as you know. And while Gulzar Khan, he is a research associate and lecturer, and he also works together as a managing editor of Taxon. They brought in some very special speakers for today, and I would like to invite them to get us started. The title is super exciting, Reading Blindfolded, the Benefits of Good Taxonomy. So welcome, Dirk and Gulzar, and welcome to all the speakers, and please take it away.
Thank you, Lucia, for the introduction. That's really nice. It's a real pleasure to be here with you tonight. For me, it's night, as you may see in the back on my window. But yeah, when we were thinking about the webinar, one thing that carried us was that taxonomy is not just a science for itself, but it has so many implications for so many important aspects of society. And one, probably the most, is breeding of crop plants. And when we are thinking about the context of breeding with taxonomy, we were thinking of so many different aspects. And when thinking of different aspects, we thought of so many different great colleagues of us who have done such great work to enhance breeding by establishing great taxonomy and all the implications these taxonomy has for different aspects of breeding. So with that, the first of our today's speaker is probably exemplifying this diversity of the taxonomy impact on breeding. And the first speaker will be Susanne Renner. And I don't think I really need to introduce you, Susanne, but it's probably everybody should know you. And looking at your publication record, I don't think how you can avoid any of Susanne's papers because there's such a diversity of different aspects from sex chromosomes, diagnoses, biogeography, up to, as I found out, real entomological papers that you now publish. So yeah, who else could start off today with introducing the relationship of taxonomy and breeding better than you? And of course, you introduce us today to the different aspects of breeding and taxonomy in the cucurbits, which have been such a great study object for you for such a long time. So thank you very much, Susanne, for kicking this meeting off, and I hand over to you now.
Thank you very much. I already opened my first slide because you said we should take 12 minutes and I will try to stick with that. So the examples I'm going to give come from domestication, from my research on the domestication of cucurbits. If there's anything we have learned from the phylogenomic studies of the past 10, 15, maybe 5 to 15 years, it's that hybridization and introgression have been much more common than was suspected. And that the traces of these two types of events, hybridization and introgression, persist for a longer time than I think all of us had suspected. And this creates problems or doesn't make naming easier because of the widespread absence of mutual monophyly between related taxa, as will typically be the case for wild progenitors of domesticated species. So my examples are going to come from Cucumis mello, Cucumis sativus, and Citrullus lanatus. The only available complete treatment of the genus Cucumis is from 1993, and in this treatment, Kirkbride synonymized 522 names under Cucumis mello, and then grouped them into just two subspecies, mello and agrestis. Molecular data, not surprisingly, showed that agrestis, which had no geographic context in Kirkbride's treatment, is polyphyletic and paraphyletic. Here you see the Asian forms of agrestis, and most domesticated high-quality melons, honey melons, are in this clade. And this is sister to an Australian species. And then we have the African honey melon clade, which again includes wild forms and domesticated forms. And together they are sister to an Australian and Indian species. What is surprising here is that China appears to play no role, and it's not for complete lack of sampling. This is surprising because the oldest seeds assigned to the genus Cucumis come from China, and they are up to 8,000 years old, and there are many, many of them. It's not just one find. Here you see some just 2,000-year-old honey melon seeds from the guts of a mummy, and they come mostly from this region, but also from more interior parts of China. So these, not the ones on the photo here, but some of the others are very small. And these small Neolithic melon seeds suggest an independent domestication of honey melons from some wild melon in China, because usually during domestication, fruit size and seed size, go up in parallel, increase. So China does indeed have a wild species of melon, and you see photos of this plant here. It's a very distinct form, species. It was named as a species by Chinese colleagues. But since Kirkbride synonymized the name that they had created under his agrestis, the name was forgotten. Nobody has used it until I dug it out. Now here you see seeds, the seeds of this wild Chinese melon. And here you see, domesticated honey melon. And here you see the seeds of the Chinese form next to the wild African form. And even half the size of that. Really distinct. So using the Latin name for this Chinese wild melon, which exists, instead of synonymizing it under this, trash basket there of agrestis, would have helped direct research towards this possible progenitor or contributor to a possible melon domestication in China. So my second example comes from cucumbers. There is a well-known orange cucumber which has been cultivated for at least 2,000 years by the Aini and Haini ethnic group here in China, Laos, and Vietnam. Here are the orange cucumbers. And here you can see the genetic distinctness of these orange cucumbers in the context of a worldwide sampling. Highly distinct. And we showed in this paper, this is in the online supporting material, that a mutation in the asparagine substitution at residue 257 is responsible for the orange pulp. There has been a name suggested for this, but it was never validly published because the colleagues who proposed this name didn't preserve any type material. So I created a molecular diagnosis and a traditional morphological diagnosis. The molecular diagnosis, of course, relates exactly to this mutation in the asparagine. And I deposited with Chinese colleagues, obviously, a traditional specimen in herbaria, in relevant herbaria worldwide. And my last example comes from the watermelons. Watermelons are super important in China, also in other parts of Asia, but especially in China, including as this snack. This snack is sold all over the country, in airports, everywhere, very easy to get. These are the Sigua seed watermelons. They have been cultivated for at least 350 years, as is known from written records. A name was proposed 40 years ago, but without type specimens, and also there are other nomenclatural problems. So I was interested in this because, well, I've done research on watermelon domestication, but also because it's so distinct and economically really important. But so far, the phylogenomic efforts do not support the monophyly of this entity. Here are the most accessions, but some are in other parts of the phylogeny. This is all just watermelon, okay? So in the absence, I have to go back. In the absence of a molecular or morphological way to recognize this, consistently and reliably, even though it's been cultivated for 350 years, it occurred to me, well, it should be named as a cultivar. Probably other people think about this. And so I read the International Code of Nomenclature for Cultivated Plants. The last edition is from 2016. And because I found several questions when trying to understand this, I corresponded with one of the co-authors, also a previous member of the IAPT board, actually very nice. And he helped me understand the situation better. Because at first I couldn't believe it, but it is true. So type material is not mandatory for establishing a cultivar name. And further, cultivar names can have reference material, including DNA sequences or genomes, as information in a database, as virtual information in a database. I have not done anything further in the direction of naming this entity because it's so unfamiliar. This approach in the Code of Nomenclature for Cultivated Plants is so different from what I'm used to. So to summarize my talk, I gave you 3 examples of the interrelationship between naming and studying domestication. The first example from the honey melon was an example where a Latin subspecies name definitely could help direct research efforts. The second example came from a well-diagnosed variety. This was the orange cucumber, which I think having a Latin name may help communicating about this economically very important form. And my third and last example from the watermelon, to me, cultivar naming is a whole different ball game, and I personally will probably not engage in it. Thank you for your attention.
Thank you very much, Susanne. That was really informative and demonstrated how difficult it is to find good names and good taxonomy for domesticated plants. Yeah, you see the applause rising here. And demonstrates really the different aspects of taxonomy and breeding. So, are there questions? I think Gulsa will organize the discussion now.
It's hard to discuss. It's hard to discuss nomenclature. Yeah.
Yeah, so maybe one question to start off. So the last example demonstrated the problems of using the code of cultivated plants. What is the relation? What is the difficulty of the cultivated plants code compared to the ones on wild plants?
To me, one big insight, maybe you all know this, but to me it was an insight. What they state right at the start is that varieties, subspecies are not to be equated with cultivars. Okay, they state as well. You can't just go from having named a subspecies or a variety and then okay, that's a cultivar. No, a cultivar is a persistently reproduced entity, and geography and type material are not relevant. It is a different world. And I really had several very informative email exchanges with one of the co-authors on this code. I also asked him whether genomic data were coming in and whether they were going to update the code and all this. And it is a very different approach from our code. It's not every 6 years, and it's it's probably unimportant how often they update, but the procedure, how it's updated, is also different.
Mm-hmm.
And the fact that type material is not required to me was like really, aha.
Yeah, so
Yeah.
that's really a change, and since there is so much variation in cultivating that, and I see Mike Bayerler here who's working with me on kale and the different types of kale that we have, and there is also a great difficulty of correlating then names with certain phenotypes.
Yes, exactly.
Yeah, there's a question by Morgan. Morgan, you want to go in and ask the question directly, or shall I read it from the chat?
Sure, I can, if you can hear me all right.
Yeah.
Great talk, Susanne. Thank you so much. Really enjoyed that. I was just curious, you mentioned at the open of your talk that one of the big surprises of the advances in plant genomics has been just the extent of hybridization and introgression and how that signature has persisted in the genomes over time of these plants. And I wasn't sure if you might be able to give one or two examples, if there are any, of hybrid origin domesticated squashes. And if so, are there any are there genomes available for both of the putative hybrid parents?
Because in cucurbita, I think there are 3 domestication wild pairs. Yeah, I would have to write to supply a written answer to your question, okay? Because I'm not so up with the latest literature on cucurbita, squashes cucurbita. And the number of papers on these economically important cucurbits is astonishing, like 6,000 papers on Cucumis mello in the past, I think, 12 years. It's unbelievable. Most
Wow,
of them, of course, it's unbelievable. I'm not up in squashes, but I would be very happy to go to my literature and check and answer.
excellent. No, that makes sense. It just really reiterates how quickly, how rapidly genomics is sort of informing us as taxonomists and how we can work together as we're flooded with this data.
We should work together, as Dirk said in his introduction, but the working together is very limited because I really try. I emailed the breeding people, and most of them are in China. It's huge. The efforts in honey melon and watermelon and cucumber less so, are huge. Okay, but penetrating that and getting that contact and telling them about these old forgotten names proposed by Chinese researchers 40 years ago is really hard, and I try.
I can imagine. As we're inundated, we're all just, I think, overwhelmed. And so it's a challenge. Well, thank you. That makes sense. Great talk.
I think there's Stefan Beck. I see the hand up here.
Okay, yeah. Stefan, then go ahead.
Hello, Susanne.
Hi.
Hi. It's nice to see and hear you. As you looked at such a lot of cucurbitaceae, certainly you found there are some native names. They are also widespread, not only in China or in South America, but some names, certainly some people hundreds ago have get in. Have you some example for this?
No, because the species I gave as examples on which I'm doing the most research right now are all in Asia and Africa. And so I'm not familiar with the languages. There are hundreds of relevant languages, as you all know. I think 2,500 languages in Southeast Asia, right? Something like that. So not all relevant for cucumbers, but yes, no, I don't have good examples off the top of my head.
Yes, I didn't know. I thought you were looking also, as I remember Michael Nee, as he.
Yes, exactly. With cucurbita, the squashes that the first person asked me about, Michael Nee had traveled in South America to find the wild relatives of various species of domesticated species of squashes. Super important work. And he also was interested in local markets and local languages, exactly as you say.
Okay.
Okay.
Yeah, great. Thanks.
Thank you.
So I think we can go over now to second talk, especially since you mentioned the 6,000 papers on squashes. I think it's even worse in the next group that we will hear about. And thank you to Susanne for the discussion. I now hand over to Aureliano Bombarely, who is a specialist for tomato genomics and tomato breeding. He has studied before at the Cornell University, and then is now professor at the Universidad de Valencia in Spain. And since the beginning of his career, he was interested in Solanaceae and especially tomato genome. And he will introduce us to the impact of genomics on the relationship between breeding and taxonomy. So Aureliano, thank you very much for giving this talk today, and I hand over to you now.
Thank you very much. Thank you very much for the invitation. Let me see. Can you see my screen? Perfect.
Yes, we do.
So, well, first of all, I would like to say that I'm not a plant taxonomist. I work with genomes. But all the work that I have been doing, I have been relying in all the plant taxonomy, wonderful work that has been done before in Solanaceae. So without that work, of course, people that we sequence genome, we will not be able to do our work. And I always think that when I wanted to talk about taxonomy, let me see. Okay, here. One sentence that I like, about naming things is this sentence from just a fantasy book from Ursula K. Le Guin that says, "For magic consists in this, the true naming of a thing." And if we don't have the names for things, so for example, in genomics, we will not be able to work with them. No? So it's for me an important thing. So today I'm going to talk very briefly, and as I said, this very briefly about plant domestication a little bit, because it's something that we work not only in Solanaceae, but in other with other crops from a genomic point of view, a little bit about the history of the Solanaceae domestication and some example of using wild species for Solanaceae breeding. So let me start with this domestication. No, for us, domestication, we need to define as the process of adapting wild plant and animal for the human use. And of course, you have many different uses. No, you can use, of course, for food, for fiber, medicines, energy, but also drugs and pigment. No, and Solanaceae are good examples of those. There are more than 2,500 plant species that has been gone under domestication. And as I said, Solanaceae, of course, is one of them. And in our lab, we are trying to study these things about selection and adaptation. No? What are the traits that we are selecting, and at the same time, that how we produce this adaptation to new environments. And maize, of course, is a wonderful example that was done for domestication. No? So you can see how the shapes change so much. No? And of course, I'm not going to go here after the wonderful talk from Susanne, but watermelon is also a wonderful example of that. So we can define different stages of plant domestication, and this is important in order to understand where these things come from and how we can use wild relatives in plant breeding. No? So one of the first steps is, of course, when we are starting to select these heritable changes, we are selecting these phenotypic changes that we call domestication syndrome. No? So it's these traits that we are selecting because they are useful for us. No, a good definition would be this suite of traits that mark the crop divergency with its wild ancestor, and this is important in order to recognize where this crop come from. No, and of course you can do many things. No, non-shattering in grasses is one example, but also reduction of some secondary compounds. That is something that happened, for example, with solanin in Solanaceae species. So plant taxonomy is essential. No, playing an essential role defining what is the wild ancestor species. No. The other thing that is important is that when we are selecting things, we are introducing huge genetic bottleneck. And the reason of using wild type is trying to overcome this problem. Why? Because with these huge genetic bottlenecks, we are reducing the genetic pool of some important genes. One of them are resistant genes. In other words, crops are usually more susceptible to pathogens. So it's the reason why we are going back to the wild. We are going back to these wild species, trying to recover these genes that we have been losing during domestication. And this is kind of this part of breeding, bringing in this four part that you can see here, these genes that we have been losing, just looking into the wild. So when we are thinking about breeding, we define the thing that is called genetic pools, and genetic pools are collections of germ plants that you can use for breeding. So the genetic pool one are cultivated varieties. So maybe you are working with one variety, but other variety may have some phenotypes that you are interested to. While genetic pool also can be some wild relatives, and we'll put some examples later with tomato. Then you have genetic pool 2 that are more different, that are not crossing so well. And then you have this genetic pool 3 that is very difficult to cross, but still you may have some interesting genes. But you need to use some very advanced breeding techniques in order to produce the hybrids. So what about the history of domestication of Solanaceae? You know Solanaceae probably better than me. So Solanaceae has between 3,000 and 4,000 species. This is from this wonderful Solanaceae species database, the Solanaceae source, and it's classified in 90 genera. But of course, many of these genera has been used and has been domesticated. Tomato and potato, of course, is the most famous one, as you may know, but you can find a very different different species that has been domesticated. For example, in the past, we were also with petunia, with eggplants, and now we also, and with tobacco too. No, we work in the lab with some nicotiana species. So the Solanaceae is a mega diverse genera with 90, the Solanaceae is a very diverse family with 90 genera. As you can imagine, and you have this division in everything that has 12 chromosomes. That very recently has been named as the dodecachroma, and everything that doesn't have 12 chromosomes. Okay. And coming back to the domestication, so the history of the domestication of Solanaceae, we need to go back many different years ago. So tomato domestication is not something that goes really far. It's around 3,000 years ago, more or less, and that was domesticated from Solanum pimpinellifolium. You have these studies wonderful studies from Jose Blanca that he explained that this moved from Peru to Mexico and then back to Peru in some ways. And that was kind of a very interesting story, no? But as I said, the message here is that was domesticated from Solanum pimpinellifolium. Once this first round of domestication, they at some point they moved from the Americas to Europe, and we have this second round of domestication and this massive breeding that happened almost 150 years ago. For the potato domestication, potatoes are much older in terms of domestication. They go around 7,000 to 10,000 years ago. Some studies put that. And it's very complex to see what is the wild species. So the most safe way to say is that it comes from this Solanum vermiculari complex. Okay, and again, once was domesticated, all this intensive breeding happened in Europe later on. Eggplant even can be older in terms of some. There are wonderful papers also, and these last genomic papers that was published in 2 years ago, they told us that was around 12,000 years ago, maybe something like that, and come from Solanum incanum, and also has some hybridizations later on. And finally, tobacco domestication is not something really old if you compare with other ones around 3,000 years ago. And two of the Nicotiana species were domesticated. One of them was Nicotiana rustica, and the other one was Nicotiana tobacco also. Okay, so let me, in some ways, just put in more information here with the breeding of some example of the breeding of these species. So tomato breeding starting, or the most intensive tomato breeding starting in the 19th century. So before the 19th century, all the tomatoes looks like this, no, with these ribs. But in the 19th century, in the US, we're starting this more intensive breeding. One of the first shapes that was selected is the one that you can see here with these ribs, no? So because in some ways was well, I guess that in some ways it was more useful because when it's completely round, it's not infected, for example, from fungi. So it's more easy to transport the tomato fruits from one place to another. And in the 20th century is when we're starting to use, well, in the 19th century and also in the 20th century, we're starting to use these wild species. No? And this is one example of these rough tomatoes that are resistant to fusarium, that is one of the fungi, and it has an integration from another wild species. So one thing that is interesting about the domestication and in tomato and happening in other domestication of other species is that we have these genetic bottlenecks, not during the domestication. So the genetic diversity went down. But then, for example, here in this paper down from the tomato breeding from the Netherlands, in the 60s, they started to use a lot these wild species. So it was when they started to increase this genetic diversity and introducing these resistant genes. So tomato, you have many different species of tomatoes. So these are the green fruit tomatoes. These are the red ones. Some of them are self-compatible, other ones are self-incompatible. And they have been using from breeding for a lot. No? So as I mentioned before, you have all different varieties for Solanum lycopersicum, but also we're starting to cross them with Solanum pimpinellifolium, the wild relatives. You have many different populations. Then also with Solanum galapagense and all the different species, some of them that will say genetic pool 3 or even genetic pool 4 because they are very difficult to cross with. Some examples of genes that has been introduced. So, for example, from Solanum pimpinellifoliums, all these resistant genes, for example, to resist fusariums or the late blight, no, to resist Phytophthora. That was good examples of integrations of Solanum pimpinellifolium, and then also from other tomatoes from other gene pools like Solanum pennellii or Solanum arcanum. No? So they are, as you can see here, no, they are trying to target this resistance of different pests. For potato, same thing. No, in some ways, they even during the 19th century and the 20th century is when all the breeding is starting with, because of course we're starting to know that you can hybridize different species, no? And you can see that in the 20th century you have different shapes and colors and size. That also these were present already in the wild relatives that were cultivated in Peru. No? They have an amazing variety. The thing that we have been learning in sequencing genomes is that, of course, you have a very complex scenario when you have wild species that are diploids, and then you have land races, some of them diploids and some of them tetraploids, and then you have the elite land cultivars that are also tetraploids. So you can see that this is something very, very complex, no? That you can combine not only different species of cultivar, but also different ploidies. One mention that is interesting is that, for example, you have some integrations in this modern cultivar from Solanum bernayi that contains, when they sequence the genome, it contains around 2.5% of the genome is from a wild species. And these produce or give to the cultivated potatoes these resistant to some nematodes, pathotypes, and also to Phytophthora. That is a major problem. But of course, you can have many, many different wild species of potato that can give these resistant genes. So let me wrap up with the examples of eggplants and tobacco. So for the eggplant, again, the eggplant breeding has been changed a lot. No? So you can see here some representation of the shapes of some eggplant from in Europe and also from I think that this is East. And then you find how they will look to today. You know, we have many, many different shapes. In the terms of eggplants, the eggplants are more resistant to pathogens. So we have not been using these wild species so intensively like in potato and tomato, but they have been using a little bit. No? So for example, Solanum torbum may not have been used, but Solanum insanum, of course, has been used for this breeding for different gene pools. You have all this wonderful work from Proen's lab that they are explaining all these things. But again, they are using this to try and produce some resistant genes that have these species. Tobacco is a more complex thing. You have the tobacco genus is diverse also, and you can have many different species, and not all of them has been used in breeding because you cannot cross them. Some of them has different chromosome sizes. Some chromosome numbers, because you have some nicotianas that are polyploids and some nicotianas that are diploids. No, we are working in our case with some polyploid nicotianas for the suavalentes, not for domestication purposes, but some of them. So here we have a table with some examples of resistant genes that has been introduced in the cultivated Nicotiana tabacum, no? And you can see that some of them has been from species that has different chromosome numbers, like Nicotiana alata. Some of them has the same number, no? So, for example, Nicotiana noctiflora, well, is the diploid version of have 12 chromosomes instead of 24. But yeah, you can see that plenty of resistant genes that have been moving from these wild species through breeding to the domesticated one. So the conclusions that I want to say, and more or less that was in 30 minutes, sorry, is that the use of these wild relative species has been essential for the breeding in the Solanaceae crops. No, without this use of these wild species, we will not have these resistant genes. And of course, taxonomy has played an essential role, no? Because if you cannot name something, you cannot use it, no? So that is an essential role. And I think that will be all. Thank you very much.
Thank you. Thank you, Aureliano. That was a great overview of the 4 major Solanaceae crops. So actually, what I take home from that is that, yes, we often think about hybridization as being just the crossing of 2 parents, and then we have a 50-50 offspring, but especially in crop plants, it's just a diversity of mixtures that we create here, which really defy any classification, actually. So
exactly, you are right. Not because I mean,
yeah,
some crops has, yeah. Sorry.
Yeah, go ahead.
No, I wanted to say that, yeah, some crops, I mean, some varieties may have, I don't know, maybe 5, 6, 7 different integrations from different wild types. So yeah, the terms of the species sometimes can be difficult, no, when you are working with crops, because it's a mix of many, many different species.
Yeah. So what do we make out of this now? Is this hybridization in crop plants something different from hybridization in the wild? Is this, yeah, what do we learn about this from regarding reproductive isolation between species? So we normally think about species being somehow reproductively isolated, but here we can easily cross apparently so many different plant species.
Yeah. Yeah, I mean, we have been crossing many of them, as I said. I think that almost all the wild species from tomato has been used in tomato breeding in one type or another.
Okay. And do we have people used different methods, or it's just simple crossing that they are just cross-compatible across the genus?
Well, no. Sometimes, I mean, you need to do some embryo rescue. So sometimes, yeah, especially when you are using more, I mean, more divergent, I mean, species that has diverged long time ago. So, for example, with some Solanum, I don't remember Solanum penelii, but some Solanums that are very far from Lycopersicum that you need to use embryo rescue.
Yeah.
Or for example, Nicotiana is very common, no? So when you are crossing Nicotiana lata with Nicotiana tabacum, yeah, you need to use embryo rescue, otherwise you are not going to be able to have anything.
Yeah. Right. But still, I mean, with embryo rescue, you get a viable offspring. So that's also very interesting that this is possible.
Yeah.
Yeah. Right. Any other questions?
Maybe in the chat.
Yeah, great talk, Aureliano. I really enjoyed that. Thank you. I just something that stuck out when you introduced yourself at the beginning of your talk, you mentioned, you don't describe yourself as a taxonomist, and it kind of got me thinking, well, giving this talk to a community of taxonomists, it made me wonder, as a taxonomist myself, a systematist, if you have any recommendations about how we in the taxonomic community can reach out to someone like yourself who's doing breeding and genomics with suggestions or ideas for taxa that might have putatively desirable traits. Um, would you ever receive an email from a taxonomist almost out of the
Yeah.
blue to say, oh, I know the species has X, Y, or Z? And otherwise, on the conversely to that, how might you identify taxa, wild relatives, etc., that you think might have something of value? Could you explain maybe a little bit about that?
Well, I mean, for us, I mean, we rely in your work, no, because we are I mean, we are sequencing things, but I mean, if you ask me, I probably will not be able to distinguish two different tomato wild species. You know, for me, the problem now is that if I don't have the sequence, I cannot distinguish anything. No, so it's a big problem for us. So in some ways, we rely a lot in this work, and I see many. I mean two-directional collaborations, no, with plant taxonomists. And indeed, we collaborate with many taxonomists in the Solanaceae community because, yeah, sometimes we sequence these genomes and we say, oh, we have found this. What is the thing that we can do now? Do you have any recommendation to name this differently? We should name. So we have this wonderful example of Nicotiana benthamiana that was because we work with Nicotiana benthamiana a lot. That a reaching studies say that you have many different populations in Australia and they are very different. And they wanted to rename all these different populations. And of course, it's something that at some point we need to afford. But the problem is that there are more than 10,000 papers now about Nicotiana benthamiana. So we still need to go back and trying to decide which is the best rule or the best way to act in these ways. But the thing that we have discovering with genomics is that indeed it's not one species, but a complex. So no, it's for sure. I mean, we rely on you guys to really put names in the right way. Otherwise, yeah, we don't know anything about putting the right names.
Fantastic. That makes sense. Great. Well, thank you. I know that was a somewhat complicated question and really appreciate your answer. Thank you. Great talk again.
Thanks.
Thank you. So there are no other questions. Let me briefly check whether there's a hand raised. Then I would thank you, Aureliano, for the talk and the discussion and the great insights on how molecular biologists and taxonomists work together in breeding. So yeah, that leads me to the third speaker of today, which is Richard Bateman from Kew now. Actually, he has been in his career at every major plant taxonomy institution in Britain, I think. From University of London to Oxford and Edinburgh to the Natural History Museum and now to Kew. So, and during all his this time, he has maybe lived two lives, one as a paleobotanist and one as an orchid systematist. I guess there would be great chances to have a webinar on paleobotany and taxonomy as well. But today I invited him especially because one story of breeding and taxonomy interaction that I came across during my PhD, which I spent for a considerable time at Kew, was the case of the orchids, Dactylorhiza orchis group and other orchid examples where there was, great interaction between breeders and taxonomists. And I thought that's a good opportunity now then to invite Richard to give a summary of what orchid taxonomists and breeders have done together to clarify the evolution of orchids and to enhance breeding of orchids. So without further ado, I hand over to Richard for the third talk of this webinar.
Okay, thank you very much for that introduction. Of course, I've moved around so much because I upset so many people. I can't afford to stay anywhere more than 3 or 4 years. However, one group of people I haven't particularly upset are the amateur growers of orchids. And I've chosen to use my 12 or 13 minutes to indicate the kind of interactions I've had, particularly with amateur growers of European orchids. You might have expected me to go straight into an orchid that we know most about, Phalaenopsis, the orchid equivalent of Arabidopsis, and talk about the industrial scale growing that takes place in areas like Taiwan and China. But the truth is I've received some useful information from those studies, but there's never been an active interaction between them. And it's the active interaction between disciplines that I want to focus on here. So this is my cast of characters. These are my 4 heroes of the 4 sections of this talk. I won't introduce them now. I'll introduce them one by one. So first up, who lives just down the road from Kew, is the late Barry Tattersall. Now, his primary reason for growing European orchids was to show them in the hope of winning prizes, of which he won vast numbers. But the way he operated was to go around Europe collecting single seed pods and then growing them aseptically, raising them to maturity. And you can see the kind of diversity that he had available in his greenhouse and his garden. And naturally, I took full advantage of such a convenient source of material. So, for example, when I wanted to, or when Paula Rudal and myself wanted to survey the detailed floral structure of these bee orchids across the genus Ophrys, the obvious place to go was not to spend a week in the Mediterranean. It was to pop round to Barry's greenhouse. And a couple of visits gave us all the major groups of species that we needed for the study. A few years later, in fact, a decade later, I was back in his greenhouse again, rampaging through his office for a second time, because we decided to apply confocal microscopy and flow cytometry to looking at the variation in genome size across a single petal, the labellum of these orchids. And there's a massive variation in genome size depending on where you are on this single petal. All of this fascinated Barry. I was always very careful to feed back to him what we were discovering. But the first time I met Barry, and the first contribution he made to my research, was when we were producing the original molecular phylogeny for this subtribe Orchidinae, which covers the majority of European orchids. So here is our phylogeny, and it's pretty well sampled at the species level and several individuals of all genera. And the reason for that is that we put a lot of effort into doing fieldwork across Europe, but also we were able to fill in the gaps, and there were plenty of them, by going round to Barry's greenhouse. But of course, the consequence of doing very early molecular phylogenetics and then applying monophyly to your results is that you cause a certain amount of disruption to the pre-existing taxonomy. And in this case, the tongue orchids of the genus Serapius survived monophyly, the bee orchids survived monophyly, the lizard orchids survived monophyly, but what had been the genus Orchis turned out to be triphyletic. And we were then faced with the decision of either lumping all of these genera together, which I think would have led to our assassination, or recognizing three portions of Orchis as separate genera by adding them to what had previously been a monotypic genus. So that was our preferred solution to the tree that resulted. There have been many trees of this group since, with much greater amounts of data in them, but the genera have survived all of those reanalyses. So you end up mixing and matching some of these species into new genera, which led to Q scientists in the late 1990s screaming, "Which is the true Orchis?" Now, of course, monographers were very quick to pick up on this information. And very soon after we had published some of these early trees, Kretschmar et al. Published a very nice monograph of what had been the genus Orchis. So unfortunately, they're publishing a monograph of a non-monophyletic taxon. But they actually very cleverly put together this diagram showing which of the species in our recircumscribed genera form natural hybrids, which is the red crosses in this diagram. And their analysis very strongly supported our division of these taxa into 5 genera. But they also reckoned that there might be a reason for splitting our narrowed version of the genus Orchis into 2 genera. And Titiker and Klein came along in 2008 and separated half of our recircumscribed genus as a new genus, Androrchis. But that really didn't fit in with the molecular trees that we were getting, which were suggesting that the true Orchis was paraphyletic relative to Androrchis, and that neither had a strong supporting branch. So I combined forces with Giovanni Scapecci and Salvatore Cozzolino in Naples. Now, they are professional academics, unlike Barry, who was a baggage handler at Heathrow. But Giovanni and Sal, I think, would admit to being amateur breeders, if not, even though they're professional academics. Nonetheless, they embarked on a massive breeding program of artificial hybridization. And they operated within the subtribe Orchidinae, and they tried 161 pairwise combinations of species within that subtribe. This is just the tip of their iceberg that I'm showing on the screen. So a phenomenal amount of work to try and work out how much post-zygotic isolation there is. But we then got together to discuss whether those data on post-zygotic isolation could be used to compare 3 different classifications of the same species. On the left here, you have what I would call the pre-molecular classification. In the middle is our molecular monophyletic classification. On the right is Titiker and Klein's attempt to break up our 3 genera within Orchis into 6 genera. And the way we tested this was to look at all of the crosses that Giovanni had produced and see what we were looking for was circumscriptions of genera where within the genera there'd be a reasonable amount of gene flow, but between the genera there wouldn't be any. So if you look at the left-hand column here, these are hybrids between Platanthera, Dactylorhizis, Hemantoglossum, Serapius and Ophrys, and they're all fully post-zygotically isolated, as you would hope. So good genera, no one ever doubted that. Then you look at the traditional pre-molecular classification, and the intergeneric crosses and the intrageneric crosses are pretty much equal. There's some leakage of genes between the two, but there's no difference between the intergeneric and the intragenetic generic, and we're hoping for a lot more intrageneric than intergeneric. And that is what we got with our molecular recircumscription of the genera. You get complete isolation for intergeneric crosses, but you only get 50% post-zygotic isolation with the intragenetic crosses. And then finally, we tested Titiker and Klein's hypothesis of Orchis and Androrchis as possible separate genera. And what we were hoping to find was that Androrchis was a good genus and that we would see complete isolation between Orchis and Androrchis crosses. What we actually found was that the crosses between Orchis and Androrchis were no better than any other intragenetic crosses. So we rejected Androrchis as a hypothesis of a genus circumscription. Now, we were rather hoping that that might stabilize the 3 genera that now represent the old Orchis. But of course, that was a hopeless wish within the orchidological community, in which there are more opinions on taxonomy than there are species. So just a year ago, we had a reconfiguration of these, what we regard as 3 species of Orchis, into 10 species, one of which is paraphyletic and one of which is polyphyletic, and several of which are, in my opinion, monotypic. Now, my third hero is Svante Malmgren. He is a surgeon who, in his copious spare time, breeds European orchids at an industrial level in his barn. Now, Svante is world-renowned because he produced the best aseptic growing medium for orchid growing, which has been taken up by the professional breeders. And he mixes sugars, amino acids with turnips, potatoes, and the very most curious use for pineapple I've ever seen. But that's his growth medium. He goes around, he collects capsules, takes the seed out, grows them up through this aseptic procedure. And in the end, he has an absolute smorgasbord of orchid tubers of many different species of temperate orchid, which he uses mainly he breeds mainly out of curiosity, and he uses these mainly for reintroduction programs. So I asked him to take a particularly close look at the genus Ophrys. The reason being that our relatively recent molecular trees like this show 9 strongly supported groups that have the kind of genetic distance between them that you would expect of a typical species of any family of plants. But for orchid taxonomists, this diagram represents roughly 450 species. There are about 200 species in that tiny little cluster over on the top left, and about 100 species in that tiny little cluster on the right-hand side of this diagram. So what we have here are 9 what I call macro species and 400 plus of what I call micro species. Now, the breeding example I use for orchis is just done by the traditional academic approach within a 3-year grant awarded cycle. So all you do is do the crosses and look at the percentage of apparently viable seed. For Svanti, you then sow them, you look at the speed of germination, you look at the speed of growth, you look at the ability to actually flower, and then you do F2 crosses and back crosses. But his favorite trick, and the one I'm most appreciative of, is when he selfs a single individual. All 4 of these examples are where he selfed a single flower to produce that level of phenotypic variation. Now, if you can store that phenotypic variation in your mind and then look at some of the microspecies that have come out of a couple of recent monographs, I think you would agree that there's more variation coming out of a single self capsule than you can see between these illustrations of supposedly different species. When he breeds 2 of the macro species together, he gets, of course, even greater phenotypic variation. And he finds that that phenotypic variation is unusually vigorous and robust, as you often have in F1s. If he then breeds the F1s together to produce an F2, the converse is the case. The growth is much weaker. The chances of getting anything to flowering size, even after a longer period of time, are much, much less. And when he does back crosses, he finds the same consequence, much reduced fertility. On the other hand, the main reason for this reduced fertility is the inability of the pollen to grow all the way to the ovules. So by taking a cross and then adding to that pollen from one macro species, he can gradually build up a hybrid that contains genetic material of large numbers of macro species. So here, the plant on the left has genes from 5 of the 9 macro species. The plants on the right have genes from 6 of the 9 macro species. And of course, his objective is to achieve all 9. I don't have time to talk about my 4th hero, John Hagar, who is a veterinarian and grows vast numbers of dactylorhizas in his spare time. But his main interest is knowing that allopolyploidy is so common in this genus. He spends all his time trying to take the 2 diploid parents and reconstruct the polyploid descendant species. Just going back to Ophrys for the final slide, in 2012, I published this diagram, which shows the number of species of Ophrys, a bee orchid, recognized over a 100-year period. You start off between 10, 20, 30, and then in the late 1990s, all of a sudden, ecologists begin describing these species, and the number rockets. And I predicted in 2012 that by 2025, we would have 450 Ophrys species. And in fact, that total was achieved 1 year early with 7 species to spare. So you have to decide whether to believe me, and this genus has 9 species, or to believe some of the splitters who believe it has at least 450. So what do I think are the benefits of the symbiosis I've had with the breeders? Well, I started off by talking about the reliable supply of research materials that they've given me, but I've gone back to the well numerous times as technology has changed. I've talked about their innovation in growing methods, which has actually fed into the professional growers. I've talked about their willingness to make requested crosses over long periods of time. They don't have to worry about funding cycles. I've talked about their constant observation of the progeny through many generations. The feedback from me to them has encouraged them to develop their own hypotheses about heredity, which has made for really interesting dialogue. The phylogenetic feedback from me has improved the cultivation for particular groups of orchids. by having successfully bred all these orchids, we've got to maximize probability of successful reintroductions. And my only disappointment for the last 30 years of interactions with these wonderful people is my failure to discourage later taxonomic tinkering, which goes on now and will go on forever. Thank you very much.
Thank you, Richard. That was really a great overview of orchid systematics in the last years and the interaction with breeders. It reminded me that actually the techniques in taxonomy and breeding have stayed the same since Linnaeus, who did these massive breeding programs.
Yeah.
And in fact, we still rely on massive breeding projects to enhance both taxonomy and breeding nowadays, and especially the interaction, and not just by professional breeders, but also by the many amateur botanists and breeders that are out there. And I could name some of these people for groups that I work on myself. So yeah.
Okay.
Aureliano had a comment or question in the chat I just saw. So Aureliano, you want to state that here?
I think that I already answered the question from the chat, I think.
I have a comment.
Yeah.
Sure, go ahead.
Wonderful talk, Richard. Seeing the name of Karel Kreutz there as having, you know, the person who published some 475 names of orchids or something, he is also active in Orobanche. You probably know this. He has published an amazing number of new species of Orobanche. Okay, sorry. Is that incredible? And then I've read many nice emails with him because he's out in the field collecting these things and then unfortunately naming them.
Yes.
A very nice person. Very nice person.
He is a very nice person, and he has an unparalleled knowledge of European orchids. I just wish he'd put it to better use.
Thank you.
Yeah, but as we've heard in the talk before, sometimes it's also important to name somehow these variations and these phenotypes. It's just a matter of whether it needs to be named as a species or whether it can be named as some other kind, but still it may be useful for further breeding if you have some kind of name to a phenotype. Yeah. More questions? I don't see any. So yeah, I think was a great overview of the different aspects of breeding and taxonomy, and the difficulties and also the chances that are provided by interacting. Gustavo, I imagine in your career you have met amateur growers. Oh, now it's gone. So, Gustavo, you want to state that yourself?
Hi. Sorry. Thank you for your talk, Richard. In your career, we imagine that you met amateur growers with some great collections that may have been acquired by ill-gotten means, not necessarily intentional, but perhaps not compliant with international law regulation. Can you give like two, three tips for the professional scientific taxonomist community to carefully collaborate and communicate with amateur growers to avoid potentially undesirable legal implications of working with plants and or amateurs who may not have followed laws as they should, but also might have plants with scientific and taxonomic importance. So how do you build this mutual trust? Thank you.
It's a difficult one for me. I'm not sure that my hands are entirely clean. All I can say is that the breeders I've worked with, I know how they operate. as you've seen, they're capable of taking a single seed capsule and producing enough, literally producing thousands of plants for conservation reintroduction programs. None of the breeders I work with sell plants or pass them on for profit in any way, shape or form. So I think my conscience is comfortable at that slightly grey level.
Mhm. Thank you.
Yeah. Yeah, it's an important issue, of course. Legal aspects and Nagoya Protocol, and I mean, in the past, especially the orchid enthusiasts have been especially almost criminal, or sometimes really criminal, when getting new orchid species. But this is something hopefully of the past, and hopefully more legal breeders are nowadays at work. But we have to be sure with that. Yeah.
No, I agree, but those are the people you're talking about. They're the ones who don't have any interest in judging whether something is a species or not. They just want to have as many as possible. So, for example, the moment a new slipper orchid is described in Vietnam from a single rock face, there's a very good chance it will be gone within a few months completely, and will find its way into some of the major collections. But those are not the kind of people I'm working with.
Yeah. Yeah, good. Any further question? Otherwise, I would say this was a very good overview of the different aspects, and I made a lot of notes on the different aspects and what to consider in the interactions. I've learned a lot, really, and I would really like to thank at this moment all three speakers to take the time and summarize their knowledge on the topic, and demonstrating to us as the taxonomics community how important it is to consider the breeders' interests in our work, and how much benefits we can get from interacting with the breeding community, whether it's amateur breeders or professional breeders. And I think this is, this is the major take-home message that I want to confer with this webinar. Whether you've been here online with us, or whether you hear it sometime later, which I think many people will do. So thanks a lot again from my side to you here providing these talks. Okay, so normally Lucia should have taken over, but I just got a message that she had to leave. So Morgan, you want to take over and give the toast?
Absolutely, I'd be happy to. Thank you again, Dirk and Gulzar, and all of the amazing speakers today. This was really an excellent webinar. I learned a lot, and really an interesting topic, something that's very different from our previous two webinars. So thank you again. I did want to ask if everyone who's here could do us a favor, if you feel comfortable, turn on your camera so that we can see everyone. And I don't know if you have a beverage nearby. I have my very large coffee, which powers me through the day. I need it today on this Friday, as it's been a long week. And we want to just take a moment to acknowledge 75 years of the International Association for Plant Taxonomy with a toast to the next 75 years, and gratitude to the amazing taxonomic community of researchers, systematists, both professional and amateur, as we've learned about, whose knowledge has driven and advanced really the foundation of knowledge about plant biodiversity, fungal biodiversity, and algal biodiversity for these past 75 years. So with that, here's to 75 years of plant taxonomy around the world, and here's to 75 more. Cheers, everyone.
Cheers.
Cheers.
So, as Lucia mentioned at the beginning of this talk, we will, of course, be continuing this webinar series. It's been very successful. We've had some wonderful talks. We do post these and make them available on the IAPT YouTube channel, so you can see our previous webinars. And this webinar will be available, the recording, in the next week or so. As we do plan for the 2026 series of webinars, we want to encourage all IAPT members to please send us suggestions or any interest that you might have in a particular topic, and we can cater these webinars so that they do address areas of taxonomy that might be intersectional, like what we had today with breeding science and taxonomy, or things that might focus more on aspects of training and taxonomy that might be really useful. So this is what we're trying to target, and this is really a great benefit for members of IAPT. So keep an eye out on future newsletters for announcements of our next IAPT webinars, and please send emails, communicate through our IAPT newsletters, and let us know how we can provide content that's going to be relevant and impactful for you. With that, thank you, everyone. Happy holidays. Have a wonderful rest of the year, and we hope that 2026 gets off to a very fulfilling and successful start.
Thank you.